Yo, what's up everyone? My name's Kashi, and in today's episode we're going to be learning all about GPCRs and G proteins. Now, if you watched our last episode, you would have seen that we established the following cell signaling framework. And before we get into this video, let's do a little review. So you have your parent cell, right? And your parent cell produces a messenger molecule. That's the first step of production. And sometimes this messenger molecule is packaged and other times it isn't. So packaging is a maybe. And eventually this messenger molecule gets released from the cell. That is the release step. And it makes its way over to your target cell. And it goes to the target cell and it produces a response, which is the next step. And then eventually this response has to get terminated to prevent it from getting out of control. And that's the last step, termination. So this is the cell signaling pathway. Now in the next few episodes, this episode and the next few ones, we're going to be looking at all about this response step. The response step is, I'm not going to lie, it can be a bit complicated. I mean, think about it. Even on paper it sounds complicated. Like how can a messenger molecule magically just go over to a target cell and produce a response? Like how does that even work? It may seem overwhelming at first, but don't worry, we're going to break it down nice and easy and we're going to go one example at a time. So let's get into it. So one of the most common ways that a messenger molecule can interact with a target cell is by binding onto receptors that can be found either outside the cell bound to the cell membrane or inside the cell in the cytoplasm. It doesn't always have to bind to receptors, but this is a very common way that it can interact with these cells by going and attaching onto them. And then when it binds onto these receptors, different things can happen. One common thing that can happen is that intracellular signaling proteins, so signaling proteins that are inside the cell, they can get activated. So when it comes and binds onto the receptor, it can activate these proteins. And these proteins can go and they can activate enzymes, which can do different shit, and that can produce a response. So again, it doesn't have to bind onto receptors, and it doesn't have to activate signaling proteins, but this is a common way how messenger molecules activate responses in cells. The thing you have to remember with cell signaling is that there's always exceptions. So you can't memorize a single pathway. You have to have a adaptable mindset. So in today's episode, we're going to be focusing on a specific type of receptor and a specific type of signaling protein. And actually, this receptor and signaling protein act together as a pair. So the receptor we're going to be talking about is called the GPCR. And the protein we're going to be talking about is called the G protein. And these often interact to create a huge variety of responses. Now, what GPCR actually stands for is G protein coupled receptor. So as the name suggests, it's a receptor which is coupled and interacts and works with G protein. So these two act as a pair. So the GPCR is a type of receptor that we would call a transmembrane receptor. Transmembrane meaning it goes back and forth across the cell membrane. It goes through the extracellular, which is the outside of the cell, extracellular part of the cell, and the intracellular part of the cell, which is the inside of the cell. And so you have your receptor, right? Your GPCR, which is bound to your cell membrane. And this GPCR receptor, it then goes into the cell, out of the cell, into the cell, out of the cell, going across the cell membrane, hence the name transmembrane receptor. And what this means is that it can interact with different things both on the outside of the cell and on the inside of the cell. So on the outside of the cell, you can have things like your messenger molecule, which can come and bind onto it. And on the inside of your cell, you have things such as G proteins, which can come and bind onto the intracellular part of the cell. And yeah, so you have these things interacting on the outside and the inside of the cell. But before we get ahead of ourselves, let's take a look at what G proteins actually are. Alright, so G proteins they're basically a type of protein, right? That is made of three different subunits. You got your alpha subunit, you got your beta subunit, and your gamma subunit. Don't ask me why they draw gamma as Y, but they call this the trimeric protein, meaning it's made of three different parts, hence the name tri. But what you'll find is that this trimeric G protein often doesn't exist alone. You can have different things or nucleotides come and bind onto them. For example, GDP. GDP can come and bind onto this G protein. And GDP basically stands for guanosine diphosphates, meaning a guanosine that is bound onto two phosphate groups, di meaning two. But GDP is not the only thing that can come and bind onto your G protein. You can also have GTP. GTP is very similar to GDP. It's still a guanosine, but instead of having two phosphate groups, it has three phosphate groups. Hence the name guanosine triphosphates instead of diphosphates. Now, at rest, right, when you have a cell and you have your G protein, not your G protein, your GPCR, your receptor, which goes in and out of the cell, on the intracellular part of your cell, you can have your G proteins bound. So you can have your alpha, your beta, your gamma subunit all bound together on the inside part of your GPCR, and you'll often have GDP. So at resting state, you often have your G protein with GDP bound to the intracellular part of your GPCR. Alright, so real quick review. You have your GPCR transmembrane receptor bound to your cell membrane, going from the outside of your cell to the inside, back and forth. And bound to the intracellular part of your cell is your G protein with your alpha, beta, and gamma subunits. And attached to that is your GDP, your guanosine diphosphates. And all is well, but then you have a messenger molecule come along. Your messenger molecule comes and it binds to the extracellular part of your GPCR. Now when it does this, this induces your GDP to leave your G protein. So your GDP goes away. And your GDP now, it's gone, right? And now it gets replaced with something new. Something we mentioned before, your GTP, guanosine triphosphates. It comes and it binds to your G protein. So now you have a new complex. Instead of alpha, beta, gamma, and GTP, you have alpha, beta, gamma, and GTP. And now comes the great rift. The G protein and GTP complex gets split in half. And so it splits into, so on one half you have your alpha subunit and you have your GTP. And on the other half you have your beta subunit and you have your gamma subunits. So it splits. So now that you have these two complexes of what was your original G protein, so you have your alpha GTP complex and your beta gamma complex, these two protein complexes can go off to activate different enzymes. So your beta gamma complex can go activate, let's just say, enzyme A, and your alpha GTP complex can go over and activate enzyme B. And these two different enzymes can produce two different responses. So let's just call that response 1 and response 2. And what's interesting is sometimes these responses can actually counteract or work against each other. But the main point is that these protein complexes activate enzymes and produce responses. And this is how the response step of cell signaling happens. This is the mystery. Now, how these enzymes produce responses is actually a bit more complicated, and we're going to get into some more examples in future videos. But for now, know this. Your GPCR protein was activated when a messenger molecule came and bound to it, causing GDP to be exchanged for GTP, causing the split in your G protein. And now you have these two different halves that can activate various different enzymes and do various different things to produce a response. So now that we've produced a response, are we chilling now? Are we good? No, of course not. Because after a response comes the next step, which is termination. These responses, they can't continue forever. They need to get terminated. And let's take a look at how this can happen. So now that you got your GTP and alpha complex going to enzyme 1 and producing a response, and you got your beta subunit and gamma subunit going over to enzyme 2 to produce a response, you need to somehow stop these responses. You can't have them go indefinitely. And in order to terminate these responses, you need an enzyme known as GTPase. And what GTPase does is it dephosphorylates GTP. What does this mean? Well, remember, we said GTP is you got a guanosine bound to 3 phosphate groups. Triphosphate, 3 phosphate groups. GTPase is an enzyme that comes along and dephosphorylates GTP, meaning it takes away one of those phosphate groups. And without a phosphate group, it's no longer GTP, it is now GDP, because, remember, GDP is the same thing, guanosine, but with 2 phosphate groups instead of 3. So it effectively transforms GTP into GDP. Now, when it turns GTP into GDP, now there's a lot more attraction between these subunits again. Because remember, when GDP was replaced with GTP, that's when these two complexes split apart. But now that we've gone from GTP back to GDP, now that attraction or that affinity, let me write that down. Attraction/affinity is very high between these complexes. So they're going to come together and they're going to recombine and form what we had originally. Your GDP, your alpha subunit, your beta subunit, and your gamma subunit. They're going to come all back together. So now that they're here, they're no longer activating these enzymes and producing a response. So you have successfully terminated these responses. So now that you've returned to your original G protein complex with your GDP and your alpha, beta, and gamma subunits, you've gone back to square one. So this can go and reattach onto the intracellular part of your GPCR. So, let me shift that over. So you have, once again, your GDP, and you got your alpha, beta, gamma subunits all linked up together. And now, so it's like a cycle, it repeats. So once again, you can have a messenger molecule that comes and binds. It replaces GDP with GTP, and then this can split into alpha and GTP and beta gamma, so 2 different compounds. These can activate different enzymes, produce different responses. Eventually, GTPase comes and terminates these responses by dephosphorylating GTP, removing a phosphate, which effectively turns it into GDP. The attraction between these two once again increases. So this ends up again with your beta, alpha, gamma, GDP, your OG subunit, and then the cycle continues. In conclusion, I hope this video was able to really help solidify your understanding of GPCRs and G proteins. In the next couple videos, we're going to be looking at different types of G proteins and how they can go and activate different types of enzymes and produce different types of responses. But in the meantime, I would really appreciate if you took a second to like and subscribe, and if there's anything that I could improve on, or if there are any sort of feedback or comments you have, please, I would really appreciate it. I know that I'm just starting off. There's lots more ways that I can improve, and I'd appreciate any suggestions or tips. Thank you guys so much for watching. Take care. I'll see y'all next time.